Microbiology, Wikipedia audio. Microbiology is the study of microorganisms, those being unicellular, multicellular, or acellular. Microbiology encompasses numerous sub-disciplines including virology, parasitology, mycology, and bacteriology. Eukaryotic microorganisms possess membrane-bound cell organelles and include fungi and protists, whereas prokaryotic organisms, all of which are microorganisms, are conventionally classified as lacking membrane-bound organelles and include eubacteria and archaebacteria. Microbiologists traditionally relied on culture, staining, and microscopy. However, less than 1% of the microorganisms present in common environments can be cultured in isolation using current means. Microbiologists often rely on molecular biology tools such as DNA sequence-based identification, example 16's rRNA gene sequence used for bacteria identification. Viruses have been variably classified as organisms, as they have been considered either as very simple microorganisms or very complex molecules. Prions, never considered microorganisms, have been investigated by virologists, however, as the clinical effects traced to them were originally presumed due to chronic viral infections, and virologists took search discovering infectious proteins. History. The existence of microorganisms was predicted many centuries before they were first observed, for example by the Jains in India and by Marcus Terentius Varro in ancient Rome. The first recorded microscope observation was of the fruiting bodies of molds, by Robert Hooke in 1666, but the Jesuit priest Athanasius Kircher was likely the first to see microbes, which he mentioned observing in milk and putrid material in 1658. Antony van Leeuwenhoek is considered a father of microbiology as he observed and experimented with microscopic organisms in 1676, using simple microscopes of his own design. Scientific microbiology developed in the 19th century through the work of Louis Pasteur and in medical microbiology Robert Koch. The existence of microorganisms was hypothesized for many centuries before their actual discovery. The existence of unseen microbiological life was postulated by Jainism, which is based on Mahavira's teachings as early as 6th century BCE. Paul Dundas notes that Mahavira asserted the existence of unseen microbiological creatures living in earth, water, air, and fire. Jain scriptures describe Nigatas, which are submicroscopic creatures living in large clusters and having a very short life, said to pervade every part of the universe, even in tissues of plants and flesh of animals. The Roman Marcus Terentius Varro made references to microbes when he warned against locating a homestead in the vicinity of swamps because there are bred certain minute creatures which cannot be seen by the eyes, which float in the air and enter the body through the mouth and nose and thereby cause serious diseases. In the Golden Age of Islamic civilization, some scientists hypothesized the existence of microorganisms, such as Avicenna in his book The Canon of Medicine, Ibn Zur who discovered scabies mites, and Al-Raza who gave the earliest known description of smallpox in his book The Virtuous Life. In 1546, Girolamo Fracastoro proposed that epidemic diseases were caused by transferable seed-like entities that could transmit infection by direct or indirect contact, or vehicle transmission. In 1676, Antony van Leeuwenhoek, who lived most of his life in Delft, Holland, observed bacteria and other microorganisms using a single lens microscope of his own design. He is considered a father of microbiology as he pioneered the use of simple single-lensed microscopes of his own design. While van Leeuwenhoek is often cited as the first to observe microbes, Robert Hooke made his first recorded microscopic observation of the fruiting bodies of molds in 1665. It has, however, been suggested that a Jesuit priest called Athanasius Kircher was the first to observe microorganisms. Kircher was among the first to design magic lanterns for projection purposes, so he must have been well acquainted with the properties of lenses. He wrote concerning the wonderful structure of things in nature, investigated by microscope in 1646, stating, "Who would believe that vinegar and milk abound with an innumerable multitude of worms?" He also noted that putrid material is full of innumerable creeping animalcules. He published his Scrutinium Pestis in 1658, stating correctly that the disease was caused by microbes, though what he saw was most likely red or white blood cells rather than the plague agent itself. The field of bacteriology was founded in the 19th century by Ferdinand Kohn, a botanist whose studies on algae and photosynthetic bacteria led him to describe several bacteria including Bacillus and Vegeto. Kohn was also the first to formulate a scheme for the taxonomic classification of bacteria, and to discover endospores. Louis Pasteur and Robert Koch were contemporaries of Kohn, and are often considered to be the father of microbiology and medical microbiology, respectively. Pasteur is most famous for his series of experiments designed to disprove the then widely held theory of spontaneous generation, thereby solidifying microbiology's identity as a biological science. One of his students, Adrian Surtees, is considered the founder of marine microbiology. Pasteur also designed methods for food preservation and vaccines against several diseases such as anthrax, fowl cholera, and rabies. Koch is best known for his contributions to the germ theory of disease, proving that specific diseases were caused by specific pathogenic microorganisms. He developed a series of criteria that have become known as the Koch's postulates. Koch was one of the first scientists to focus on the isolation of bacteria in pure culture, resulting in his description of several novel bacteria including Mycobacterium tuberculosis, the causative agent of tuberculosis. While Pasteur and Koch are often considered the founders of microbiology, their work did not accurately reflect the true diversity of the microbial world because of their exclusive focus on microorganisms having direct medical relevance. It was not until the late 19th century and the work of Martinus Bijerinck and Sergei Winogradsky that the true breadth of microbiology was revealed. Bijerinck made two major contributions to microbiology: the discovery of viruses and the development of enrichment culture techniques. While his work on the tobacco mosaic virus established the basic principles of virology, it was his development of enrichment culturing that had the most immediate impact on microbiology by allowing for the cultivation of a wide range of microbes with wildly different physiologies. Winogradsky was the first to develop the concept of chemolithotrophy and to thereby reveal the essential role played by microorganisms in geochemical processes. He was responsible for the first isolation and description of both nitrifying and nitrogen-fixing bacteria. French-Canadian microbiologist Félix de Herelle-Co discovered bacteriophages in 1917 and was one of the earliest applied microbiologists. Joseph Lister was the first to use phenol disinfectant on the open wounds of patients. The birth of bacteriology. The branches of microbiology can be classified into pure and applied sciences, or divided according to taxonomy, as is the case with bacteriology, mycology, protozoology, and phycology. There is considerable overlap between the specific branches of microbiology with each other and with other disciplines, and certain aspects of these branches can extend beyond the traditional scope of microbiology. While some fear microbes due to the association of some microbes with various human diseases, many microbes are also responsible for numerous beneficial processes such as industrial fermentation, antibiotic production, and act as molecular vehicles to transfer DNA to complex organisms such as plants and animals. Scientists have also exploited their knowledge of microbes to produce biotechnologically important enzymes such as TIC polymerase, reporter genes for use in other genetic systems, and novel molecular biology techniques such as the yeast-2 hybrid system. Bacteria can be used for the industrial production of amino acids. Corynebacterium glutamicum is one of the most important bacterial species with an annual production of more than 2 million tons of amino acids, mainly L-glutamate and L-lysine. Since some bacteria have the ability to synthesize antibiotics, they are used for medicinal purposes, such as streptomyces to make aminoglycoside antibiotics. A variety of biopolymers, such as polysaccharides, polyesters, and polyamides, are produced by microorganisms. Microorganisms are used for the biotechnological production of biopolymers with tailored properties suitable for high-value medical applications such as tissue engineering and drug delivery. Microorganisms are used for the biosynthesis of xanthan, alginate, cellulose, cyanophycin, poly, levain, hyaluronic acid, organic acids, oligosaccharides and polysaccharide, and polyhydroxyéchanotes. Microorganisms are beneficial for microbial biodegradation or bioremediation of domestic, agricultural, and industrial wastes and subsurface pollution in soils, sediments and marine environments. The ability of each microorganism to degrade toxic waste depends on the nature of each contaminant. Since sites typically have multiple pollutant types, the most effective approach to microbial biodegradation is to use a mixture of bacterial and fungal species and strains, each specific to the biodegradation of one or more types of contaminants. Symbiotic microbial communities confer benefits to their human and animal hosts' health, including aiding digestion, producing beneficial vitamins and amino acids, and suppressing pathogenic microbes. Some benefit may be conferred by eating fermented foods, probiotics, or prebiotics. The ways the microbiome influences human and animal health, as well as methods to influence the microbiome, are active areas of research. Research has suggested that microorganisms could be useful in the treatment of cancer. Various strains of non-pathogenic Clostridia can infiltrate and replicate within solid tumors. Clostridial vectors can be safely administered, and their potential to deliver therapeutic proteins has been demonstrated in a variety of preclinical models. Branches. Applications.